Membrane proteins are the molecules that are attached to or embedded in the plasma membrane. And we are worried about those because those are the molecules that are going to actually move the important products that a cell needs across that membrane. So these proteins help determine the membrane's function, whether that is simply exporting waste or if it is exporting a certain product that is important that that cell specifically makes. So there are two major types of proteins. One are peripheral proteins. These are proteins that are loosely bound to the surface of the membrane. I'm not going to say any more about those. I mentioned them at the beginning. Our book doesn't go into detail about what they are. We are interested in the integral proteins. So integral proteins penetrate the hydrophobic interior of the bilayer. So these might be proteins that are only embedded partway, or they might be transmembrane proteins. These are proteins that cross the entire membrane. Now, just like the phospholipid bilayer that is amphipathic, these transmembrane proteins must also be amphipathic, because this region here that is embedded in the hydrophobic region has to also be hydrophobic. And this region here that sticks out into the aqueous solution surrounding the cell or inside the cell must also be hydrophilic. So these proteins have to be amphipathic as well. And they help to regulate and facilitate transport of individual molecules. Now these need to be small molecules. They are proteins, which means they can't transport something as large as a protein. So we're talking about transports of single molecules, say a single glucose or a single amino acid, ions like chlorine, small molecules like water or carbon dioxide. We have other means of transportation that will bring in those large molecules, something protein or bigger. So these proteins help regulate transport across the plasma membrane. And transport proteins allow molecules to avoid contact with that lipid bilayer. So if the molecule happens to not be hydrophobic, it can still move. And most of the biological molecules that we need to get into and out of the cell are hydrophobic because they contain a lot of oxygen and nitrogen, if you remember that from chapter 3. So channel proteins provide a hydrophilic tunnel with unrestricted movement. So if it's hydrophilic and it fits, it'll go right through. Aquaporins are specifically a channel protein for water. So water is really important for the cell, right? Just like it's important for our bodies. We have to get a lot of water into the cell. Now, carrier proteins are a bit more specific than channel proteins. They change shape to allow specific molecules to pass. Now, those carrier proteins often require energy because we've got a shape change happening, which leads us to two big broad types of transport, which is passive transport, where there's no energy required, and active transport, where there is energy required. And the remainder of our chapter is going to be focusing on the difference between those passive channel proteins and those active transport carrier proteins. Our passive transport mechanisms include diffusion and facilitated diffusion. So this diagram on the bottom shows character drawings of the two different types of transport. On the left side, we've got passive, and on the right side, we've got active. So simple diffusion means that molecules will move straight across a membrane, right through it, and will do so freely. There's no restriction there. Those are going to be your hydrophilic molecules. They're usually really small. Then we also can have facilitated diffusion. Facilitated diffusion means that we require a protein to help move that molecule. So we might have a channel that little molecule can move right through, or we might have a carrier protein, which means that a specific molecule is going to bind in that protein and then cause a shape change, which releases it on the other side. So a carrier-mediated transport can actually be active or passive, and we'll talk about how that can be in just a minute. In active transport, we might have carrier proteins. So these work the same way in that they bind a specific molecule, change shape, and then basically open up to the other side. So I like to think of this as like a revolving door or a turnstile. It's not free movement, but when you cause a shape change, you can get through. We can also have pumps. Usually these are ion pumps, so they create a charge. And we can also have co-transport. This is where you have two different molecules transported at the same time. So let's talk a little bit more about passive transport. In passive transport, we have the movement of any substance across a plasma membrane, or really across any surface, but it does not require energy. So there is no energy investment. Instead, molecules move along a concentration gradient. So they move from where they are highly concentrated to areas where they are less concentrated. Our two mechanisms for this are diffusion and osmosis. In diffusion, we have movement of particles of any substance, so any substance can move by diffusion, movement of particles of any substance, so they tend to spread out into available space. Now this can be across a membrane, like a cell membrane, or it can simply be through a solution. So I like to think of diffusion as people leaving a Cardinals game, or people leaving a Blues game, or a concert at Enterprise Center. Everybody's inside in a really high concentration, and at the end of that concert, everybody wants out. They're going to go out the doors, and they're going to spread out into areas where they are less concentrated, into available space. So these molecules are going to move along a concentration gradient. So without any other forces, nothing physically pushing them out, a substance is going to move down a concentration gradient. What this means is a substance will move from an area of high concentration of that substance to an area of low concentration of that substance. Now this is substance specific. So diffusion of one substance does not affect the diffusion of another. So as a quick example, let's say we have a large beaker and we put a drop of dye into it. We put some red dye into it. Now, those molecules in that red dye are going to start hitting each other. And if you don't shake it up or anything, those red dye molecules are going to eventually spread out until your whole solution is red. And that's because they are moving from the droplet where they were highly concentrated out into that available space in the water that makes the solution. This image here shows the overall movement of molecules. Now in this case, we've got a membrane. So there's not complete free movement. Those molecules have to move. But in diffusion, these molecules of dye are highly concentrated and they're actually moving around at the molecular level. So even if you aren't shaking them, they're moving and they're bouncing off one another because they're highly concentrated. And so as they move, they're going to start bouncing through this membrane. And they're going to continue to move, and in the areas where they are most highly concentrated, they're going to continue to bump into one another. And they're going to continue to spread out until we reach equilibrium on both sides. That means that we have equal concentrations on both sides of the membrane. Now, keep in mind that with diffusion, we don't have to have a membrane. So these molecules might not be moving across that membrane, but they're still spreading out into available space. If we have more than one molecule, each molecule is going to move based on its own concentration. So if we have a dye on the left, the yellow dye, it's going to spread out away from the high yellow dye concentration. And that purple dye on the right is going to spread out from the purple dye concentration until we have equal distributions of both the yellow and the purple dye. Now, you might be thinking, well, what's happening to the water at this point? We've got the dye. Is the water standing still? No, water can move as well. And the special situation of the diffusion of water is called osmosis. In osmosis, water moves across a membrane. Now, this is important. Part of the definition of osmosis is the diffusion of water, but it's also that water diffuses across a membrane. This is determined, how the water moves is determined by the solute concentration across the membrane. So water, because this is diffusion, it is still diffusion, water moves from areas with a high water concentration, which seems kind of strange to think about because we usually think about the concentration of the solute. But if we're thinking about the definition of diffusion, where a particle moves from high to low concentrations, water must also move that way. So water moves from a high water concentration to an area of low water concentration. Now, the other way that you can think of this is that water moves from areas of low solute concentration to areas of high solute concentration. Because if we have a lot of water, we don't have a lot of solute. If we have a lot of solute, we don't have a lot of water. It's kind of a relative situation. But what is really important here is that water is what is moving. So water is moving across a semi-permeable membrane, and that solute is stuck. The solute is not moving. So here's a picture to demonstrate osmosis, the diffusion of water. So this U tube on the left shows solutes, sugar molecules, dissolved in water. And on the left, we have a low concentration of solute, which means we have a high concentration of water. On the right-hand side, we have a higher concentration of solute, which means we have a lower concentration of water. Now, we said that water can move across a semi-permeable membrane, but the solute does not. So the water is going to move from where it is highly concentrated to where the water has a lower concentration. And it's going to do that until there is an equal concentration on both sides of the membrane. And that's really important. Eventually, the net flow, whether we're talking about diffusion or osmosis, is equilibrium. So molecules, in this case water, but we could be talking about substances as well, will continue to move across that membrane, but there's no net change in the balance of the concentrations. So keep in mind the difference here. Diffusion is the movement of any substance. Osmosis is the diffusion of water. So when you're thinking about what's going on, you need to think about, is there a membrane, and what is or can actually move in this situation? So I will leave you with this image here. I want you to think about what is moving, how it's moving, and whether or not this is osmosis or diffusion. So let's talk about how cells regulate the water that's moving in and out. So we said that water can and does move across a plasma membrane. And if we're talking about movement of solutes and water, you might be thinking that there are solutes inside of a cell that might actually cause that cell to take up or release water based on those solutes. And you would be absolutely right. So water balance in a cell is called tonicity. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. And what we mean by this is the solution that surrounds a cell. So there's our little cell with a nucleus, and there is water surrounding that cell. And both inside the cell, there are some solutes, and outside that cell, there are some solutes. But the surrounding solution is going to cause the cell to either gain or lose water, depending on where those solutes are most highly concentrated. So solutes in the surrounding solution, and that's really important, in the surrounding solution, cause water to diffuse, or, because we're talking about water, to move by osmosis into or out of the cell. Now, I'm going to give you some terms. Your best bet is to actually sit down and memorize them right now. Now, these terms can confuse students because they're not always sure what situation they're talking about. When I refer to these situations, I am talking about the characteristics of the surrounding solution and the effect that they have on the movement of water. Our first solution is isotonic. So iso means same. That means that the solute concentration outside the cell is the same as the solute concentration inside the cell. So solute concentrations in and outside the cell are equal. Now remember, if concentrations are equal, we will still have movement, but that movement balances in and out. Now this also means that water concentrations are equal inside the cell and outside the cell. So what happens? Well, water is still moving. Remember, we're talking about the movement of water. This is osmosis. And we're talking about a semi-permeable membrane, which means solutes aren't necessarily moving. So in osmosis, water is going to diffuse across the membrane, but at the same rate in both directions, because concentration of solutes the same on both sides and water's the same on both sides. So this image here shows two different types of cells. We've got a eukaryotic cell, a mammalian cell, animal cell, blood cell. Let me get the right word there. We've got a blood cell, and on the bottom we have a plant cell. Now, these arrows indicate water's moving in and out. Isotonic is what you want for a healthy blood cell. You don't want it to lose water, you don't want it to gain water. Plant cells actually are not best in isotonic situations. There's actually a better situation for them. Now, keep in mind that plant cells store water in that central vacuole. So water can move in and out of that central vacuole based on solutes. And the plant will actually store water, and it does this on purpose. So if we're a plant cell, and we're in an isotonic situation, the plant cell is actually flaccid, which means a little bit limp. And you don't really want that in a plant cell. Our second situation is a hypertonic situation. Now this means that somewhere there's going to be a higher concentration. Hyper means higher or greater. Now remember, tonicity refers to the surrounding solution. So the solute concentration outside the cell is higher than inside, which means the opposite is true for the water. The water concentration is lower outside and higher inside. So pause for just a minute and think about how water is going to move. Hopefully, you came to the conclusion that water is going to diffuse out because water is at a lower concentration outside. So water wants to move from the high concentration inside the cell to out of the cell. So water diffuses out, causing the cell to shrink. We don't want our red blood cells to shrink. This is a bad situation. If our blood cells are not full and nice and plump, they're not carrying oxygen. And this is really the same for any type of cell. If they're in a hypertonic situation, they're probably going to lose water. They're probably going to shrink and they're not going to be able to function. They're going to die. The same is true for a plant. So in this case, our hypertonic situation has caused water to move out into the surrounding solution where the solute concentration is higher, and the plasma membrane has pulled away from the cell wall. You can probably guess again that this is not healthy for the cell. This is called plasmolysis, or the cell is plasmalized. So we've got one more situation. That is our hypertonic situation. And in a hypertonic situation, you probably guessed that solute concentration is lower outside the cell. Than inside. And so water is the opposite. Water concentration is higher outside than inside. So what is going to happen? You probably already guessed it, that water diffuses in. Because that water is in a high concentration outside, it wants to move to where it's at a lower concentration. So water is going to move into the cell. Now this is going to cause the cell to swell. You can imagine that if this is a cell in your body or another eukaryotic organism, this is probably not a good situation, because once you lyse your cell or break your cell open because that water rushes in, basically it's like a water balloon that's too full, and eventually it's going to pop and it's going to spill all of its contents into the surrounding environment. There's no recovering from popping, just like there's no recovering from popping a water balloon. So if the cell swells too much, it will burst. But this is actually a good thing for plants, and that's because of turgidity. It's a hard word to say. So in a hypotonic situation, water is going to rush into the plant cell, and it's going to fill up that central vacuole. Well, we've also got sort of a backup plan, and that is the cell wall. And that cell wall is going to exert turgor pressure on that cell. So there are ways to control water balance, and this is called osmoregulation. In plants, cell walls help to regulate changes caused by tonicity. And if you are in a hypotonic situation and you are a plant cell, you are actually quite happy. So turgor pressure is where you get cell walls pushing back on water pressure. So the plant cell, and I'll go back for just a minute, can only fill up so much until the cell wall limits how much water it can take in. And then it's not going to take any more water, but it can't burst because that cell wall is containing it. So you actually want plant cells to be turgid or very firm. And when you have a happy, healthy plant that's standing up straight and its leaves are full and supporting themselves, that's a healthy state for your plant. So the solute concentration in the soil that is surrounding your roots, where we're taking up water, is actually really important in helping to regulate how full those plant cells are. Now, flaccid cells, we saw that in the isotonic situation, are not great. This is what happens when your plant starts to wilt and the leaves are all droopy. It's because they need some water, basically. They need water to fill up those central vacuoles. And then, of course, we had that hypertonic situation for the plants that was really bad. That's going to shrink the plasma membrane. It's going to stretch away from the cell walls, and your cell is going to die. So you definitely don't want that for your plant. Animals also have ways to control their water balance, so they use osmoregulation as well. So these cells, if you are in a non-plant eukaryotic cell, something without a cell wall, you don't have that outside structural support, at least not a really strong structure. And so there are adaptations to combat this. So one adaptation is the control of those solutes and the water balance, called osmoregulation. Now, this organism here is a paramecium, It is a eukaryotic cell, but it is single-celled. And if you look here right along the edge, they have these little cilia, and that's how they move around in their environment. And they beat like oars. They're kind of cool little guys. And they have a central vacuole. So this is a vacuole for water storage. And they can combat the movement of water, so they can counteract movement of water by pumping water in or out of that contractile vacuole. So that contractile vacuole is also working as storage for water, but the water can be moved depending on the needs of the cell. So if water is rushing into the cell, it can be pumped out. Now, if you're familiar with boating, this is kind of like a bilge pump. You get water coming in from the lake around you, and the bilge pump pumps it back out so you don't take on too much water.